was like, I told you not to. Oh, yeah, let's early. That might give. Alright, hello everybody, thank you for coming. Derek is always one of my favourite guest lecturers because we always have massive issues trying to get his computer to talk to Dick's. And last year actually we ran it off his computer and it didn't record. So I think we've managed to work it out, so it's all good. I'm going to introduce him in a minute, but I've got 3 things of housekeeping that I have to make sure you do. Tomorrow we're doing cell culture. We are not going to be able to put everyone through the first round of the hoods, so we're going to have 2 rounds, but still come because we actually need to, get you all through. It takes about an hour, so we should be done in 2 hours anyway, so make sure you come. The tech staff have asked anyone who's leaving their bags on the floor outside the labs, instead of putting them in a locker, please don't do that. They wheel trolleys around and it's a tripping hazard for them. So pop it into one of the lockers. Outside our lab, if you run out of lockers, there's actually lockers on the other side of the lift, sort of diagonally across, so you can go and use those lockers too. We are probably going to try and use both labs. So we might be able to get through everyone really quickly if we can get the other lab, but I haven't had that confirmed. With your primers, I am still doing them, but bear in mind, some of you have been receiving feedback from me, and so you'll know it's not simple feedback. It is actually, I look at everything and I write out an email, and sometimes it takes me an hour to work out what's happened. So if we add that up for 66 or 64 students, that's 64 hours. So be patient, all right? If I haven't responded to you probably by next Tuesday, for me it'll be too late, but just send me an email and say, hey, I still haven't got a feedback. I am working on it though, so all right. Now, I get to introduce Derek to you. So Professor Derek Richard is the leader of the Cancer and Ageing Research Program at QUT, but you'll be able to still tell from his accent that he is not from Australia originally. He's actually from Scotland. Yeah. I can't do it when I'm drunk either. But anyway, he's from Scotland. He did his PhD over at the University of Dundee and then came out to Australia as an ARC fellow, I believe, and went to QIMR and worked there until we poached him to QUT. And Derek has an interest in genome instability, which is one of the hallmarks, DNA repair, which is one of the enabling characteristics of energy sheets. And then he's going to tell you all about the work that they do. Quite a big program of people, lots of researchers, and has spun out 2 or 3 companies now.
4.
I can't keep up. 4 companies. So Derek is, an entrepreneur, a scientist, a great speaker, if you can understand what he's saying. So Good luck.
Yeah, hopefully, there is a professional. There's a seat down here if you wanted to use that. There's also 2 over there if you.
Is anyone
if you're
on
comfortable
the floor?
there, if you're comfortable there, just stay there. So great to be here. I do this talk pretty much every year for the last 4 years or so. I think it's maybe more than that, yeah. And probably the biggest class I've seen so far. So great seeing you.
Well done.
Yeah.
Good guys.
So Sally kind of introduced me and what I do. This is one of these lectures. Just stop me and ask me at any time, right? Any questions whatsoever. Just put your hand up. Has everyone seen this building up there? Kelvin Grove campus. Yeah, that's formerly the Institute of Health and Biomedical Innovation. I still call it that to annoy everyone. But this is where my lab is. We have not so big anymore. We have about 16 people or so in our lab just now working. So if anybody ever wants to do, I think you may be too late for Wales because you're all in your 3rd year, but if anybody wants to do an honours project or MPhil, we have all those positions. And these are just some of our lab and our lab website if you want to find out more about it. So a photographer did a great job at making us all the same height. So you can see me there in the front. I'm next to Milla, and Milla is like 5 foot 4 inches tall. Same height as me there. So that's our lab. You can go on our website. We're called CARB. being from Scotland, we think of carp as being this, the koi carp, the $1,000 goldfish. I didn't realise Australians hate carp, so so anyway, we stuck with that. So I just had a look before I came, a new slide. So we've done a few students in the time since 2015. I came here in 2011, 2015, 32 completions in PhDs and MPhils, 150 odd papers, 22 million in income. I'm just part of a. I don't get any much of this money, but the CRC was $238 million. And I started all those companies over the time. So you get to have fun as a scientist. You get to try and bring your inventions to the public, and the only way really to do that is to spin out a company. And Sally has done the same as me. She's bringing some of her technology through a company.
I only have one company though.
Yeah.
I'm not an overachiever.
My last one, I'm going to say my last one, the Deer Skin, is a bit of a hobby. I did a fundraising event with, or not event, a skincare company came to me a few years ago. So I'm interested in aging, that's what drives everything we do. And they came to me and said, will you help me develop a skincare range? And I said, all right, yeah. And they said they'll give us, I think it's like $3 for every bottle they sold. And so I didn't really believe in the industry, but I did it anyway. It turned out it really worked. So, but unfortunately, that company went down in COVID times. They were an online company only, and anyway, so I after being tested by a few dozen customers, I decided to open and start making it myself. So I converted a room in my house into a lab, and I manufactured the weekend skincare products. So the Deer Skin, if you want to try it out. All right, so like aging's really hard to get funding for because it's not a disease. You know, it's a natural process, so you can't really write to the National Health Medical Research Council and ask for money to fund aging research, and you can't write to the Australian Research Council either. So we have to pick diseases, and you know, we know cancer is you know, it's still a major problem. We're not dying from infectious diseases so much around the world. We're living a bit longer in general in every country. And as a result, we start to get hit with the aging diseases. And, you know, we might think that smoking, etc., is the #1 cause of cancer or lung cancer. It's not really. It's actually age. Age is the #1 risk factor of cancer. We know children get cancer, but it's a very, very, very small percentage of children. I might have a graph of it, but if you look at a graph, you can't really even see it on a graph. And their children's cancers are quite different in the etiology as well from our cancers. So it's primarily an aging disease. And when you start looking at other diseases that plague this our community, you start looking at osteoporosis. #1 risk factor is age, dementia, age, Parkinson's, Alzheimer's, old age, sarcopenia, age, etc. So we're trying to understand how we age and why we get the diseases. Is there anything we can do to stop that? And to kind of prove that, you know, age is the major factor. If you survive to old age, that means you don't get run over by a bus or something when you're younger. Your mathematical chance of getting cancer in your lifetime is about 70%. Now, in Australia, it's slightly worse than that because when they calculate these numbers, they exclude skin cancer. And therefore, I would say in Australia, probably most of us will get cancer at some point in our lifetime. And so you then have a question, is it a disease or is it a normal pathology? Is it just part of our life cycle? Are we meant to get cancer? Are we meant to get dementias? And are they realistically and depressingly just mechanisms that our selfish genome has developed to get rid of us and allow our children to survive? So obviously, before modern medicine, your chance of surviving cancer was pretty low, and the same with all the other diseases associated with aging. So what causes cancer? Cancer is caused by changes to your genetic code during your lifetime. So as we get older, our genetic code accumulates more and more mutations. Your age as long as I'm mature students here, you're all doing pretty well in coping with the amount of DNA damage that happens to your genome every day. There's about 30,000 damage events happen to the genetic code in every single one of your cells every day. And these mutations can ultimately, if not repaired, change genetic code. And when you lose genetic code in certain certain genes, then you can ultimately end up developing diseases. And some of us are not all born. We're not all born equal. Some of us are born with. We're all humans, right? We're all the same species, but we all look quite distinctly different. More different, I guess, than most other animals do. And that's because we don't all have exactly the same genetic code or the epigenetic printing on that code. So that makes us look how we are. Some of us are unfortunately born with defects in the DNA repair proteins. Great example, who's heard of the BRCA genes? Hopefully by this point in your biomedical careers, most of you have heard of so BRCA1 and BRCA2 are DNA repair proteins. They're found to be associated with breast cancer and ovarian cancer, and to such an extent that if you're a carrier of BRCA, it's pretty much advised that you have your children early if you're going to have children, and then have a double mastectomy and your ovaries removed. And that's because when you were born with, we've got 2 copies of every gene in every one of our cells. Some of you will be born with only 1 copy of BRCA1 or BRCA2 in your genome, and there'll be a mutation in one of the other one, which makes it unfunctional. And your chances then of getting breast cancer and ovarian cancer, approaching 100%. Not quite 100%, but approaching 100%. And you can spot these families because you'll see that who's head of Lara Croft, is it? Not Lara Croft. Yeah, Lara Croft, yeah. Yeah, Tomb Raider. Yeah.
Angelina.
Angelina, that's Angelina Jolie.
We all knew who he was.
Yeah. Angelina Jolie, so her mother died of breast cancer, and her grandmother had died of breast cancer, so she decided to be genetically tested for BRCA and found out that she wasn't that. And so she had, in her early 30s, she had a double mastectomy and her ovaries removed. The other one is Pierce Brosnan, and we know Pierce Brosnan, James Bond, for 2 or 3 movies. So his wife unfortunately died of ovarian cancer, and then 5 or 6 years ago his daughter died of ovarian cancer. And so they were a line of carriers. So genetic testing is a great one. One of the companies I did start was a company that genetically tests children to see if they've been born with defects in the DNA repair proteins or other proteins. So mutations over.
Yeah. The question.
Oh, yeah.
Do we know why the BRCA genes specifically target breast and ovaries?
So BRCA1 and BRCA2 are they're involved in the repair of double-strand breaks within your genetic code. So the other factor that you can get a mutation is ATM, which is another factor. It's a kinase involved in the exactly same process. I don't understand the biology behind it, but you'll find that a lot of cancers are hormone related. And if when estrogen enters the cells, estrogen is broken down to 2 byproducts, and one of those byproducts actually causes double-strand breaks. And I don't know if that's a deliberate or just a thing. We had a paper in Molecular Cell a few years ago describing it. Men that prostate cancer, women ovarian cancer. Men also get breast cancer, but obviously there's only about 4% of all breast cancers are male. Yeah. So that's if you imagine that red, there is 2 bases of the 3.5 billion letters of genetic code that have been changed. And that cell may, if it's not repaired, maintain that loss of genetic code. And as the cell moves on, that can then, other mutations can occur. And those other mutations combined with that one can ultimately lead to cancer. But ultimately, these mutations lead to cells getting unfit as you're getting older, and this causes a chronic inflammatory state. The cell knows that there's DNA damage happening, it's not repairing it. So I'm not sure if I'll carry on talking, and then hopefully I've got a slide. So, DNA damage is happening all the time. DNA sits within the nucleus because it hates oxygen. DNA and free radicals don't go together. DNA oxidizes extremely easily. And much of the 30,000 damage events every day are actually oxidation of bases within our genetic code. But there are other ones that are a bit more severe. Double-strand breaks, even if you don't repair a single double-strand break, that cell will die because it's lethal. Your chromosome will fall. That chromosome where that double-strand break lies will be ripped apart during mitosis, and then you'll lose the chromosome, or big chunks of it. As I say, some of us can be born with repair defects, and that can result us in getting genomic instability early in life, earlier than you should in life, and therefore the risk of cancer starts to increase. So the average onset of cancer and diagnosis of cancer in Australia is about 63 years old, slightly different for men and women. And people who are born with repaired defects, you just move that forward about 20, 30 years because the risk is so much. But you can also make lifestyle choices that will make you age faster. Smoking is a great example. Smoking, we know, is associated with lung cancer. The epidemiologists found that almost everyone who gets lung cancer are smokers, and therefore there was a link. And for many years, the tobacco companies argued that epidemiology is classed as circumstantial evidence. It's not true evidence that something happens. So they end up having to get beagles to smoke cigarettes to show that it actually happens in biology if you animals that smoke.
Got a question at the back?
Yeah.
Yes. What about vaping? Is it just as bad as smoking?
Vaping? No, right, 100%. Vaping has nowhere near the same risk factor of lung cancer as smoking. Vaping is a bit of a Russian roulette, though. So certain people, and it can be your first ever vape or your thousandth vape, can initiate a cytokine storm in your lungs, and that will kill you very rapidly. You're lucky you live in Brisbane. So to survive a vaping cytokine storm, you need to bypass your lungs. And there's only one lung machine in Queensland that can do it. And basically they go in through your neck and completely bypass your lungs until your lungs have a chance to recover. It's a bit of a Russian roulette. People die every year from vaping. And usually younger, older people don't seem to initiate the storm the same as younger people. The other one you can get is popcorn lung, which is basically a lifelong debilitating. It's like, COPD. Yeah, it's like getting COPD when you're like 16, 17, 18. So it doesn't cause cancer, but vaping is kills people, and very suddenly, very young. Yeah. So but if you're going to have a choice of smoking or vaping, vaping's as far as cancer goes.
Don't do what I
Don't do it. Yeah. Sunlight obviously is another one. So I should say smoking causes lung cancer, right? Causes every type of cancer, increases your risk of every form of cancer. In fact, all it does is makes you age faster. It contains over 60 chemicals that have been shown to bind to DNA and damage DNA, and our body was not designed to cope with that much damage. Now, we can cope with 30,000 damage events today, but if you start adding smoking to it, so if you smoke, I'll finish my fact and then I'll go on to your question. If you smoke, you will die 15 years younger than a non-smoker on average, and you'll die of everything from heart attacks, strokes to,
How about second?
Before you ask, can you guys do me a favour, because I am still learning your names. Can you say, my name is blah blah, and don't say blah. Just say
My name is Zara. How about secondhand smoking? How would that factor in? Like, you're not directly smoking, but you're always around someone who is.
Yes. Secondhand smoking, there is evidence it happens, and it does cause cancer. If you are a non-smoker and you get lung cancer, your probability of surviving is worse than a smoker. Yeah. And it's not because the disease that you get is worse, it's because when you go to a GP with a cough as a non-smoker, he'll give you antibiotics, he'll give you maybe steroids, and he'll wait for weeks and weeks and let that cough run out, and then they'll eventually. I have a friend, his mother died, and the doctor didn't give her an X-ray for a year. She had this cough for a year and kept on going back. Gave her an X-ray a year, and she had stage 4 non-small cell lung cancer, and passed away about 6 months after X-ray. Yeah. So secondhand smoking, especially in childhood, definitely linked, and worse prognosis, but not because the disease is worse. It's just because diagnosis is terrible. Yeah.
Okay, Caleb, and you said the 30 chemicals. Was that specifically nicotine cigarettes or
Anything made with tobacco? Nicotine's fine; doesn't do anything. Yeah, it's everything else is in tobacco. Yeah.
So those patches are okay.
Yeah.
Is it nootropic effects that can come with nicotine?
Yeah, look, I don't know about the other effects, but certainly in cancer, if you're looking at cancer, they don't have a risk factor. Yeah.
Oh, that was going to be my question.
All right. Okay. Sure. All right. So obviously, sunlight is another one. Some of us in this room come from ancestry that lived close to the equator. Other ones, like myself and Sally, come from Northern Europe, where there's not very much sun. And therefore, if you're close to the equator, you evolved and developed to have darker colour skin because the pigment filters out ultraviolet light and protects your genetic code from the UV. We, however, are not the case. There are some interesting facts with sunlight and cancer. Farmers do not get melanoma. Very low incidence of melanoma in farmers. It's office workers who get melanoma, and the reason is that if you work in an office and then once every couple of months you head up to Noosa, go to the beach, your trunk and melanoma is most common on your trunk or parts of your body that have been covered. So you take your top off, your milk bottle white, you get smashed with UV radiation. Your body's quite clever; it stops making proteins that it doesn't need. You're sitting in an office, so why do I need to have the basic scission repair pathway and the long patch repair pathways, nucleotide excision repair pathways that repair that kind of damage? So your cells will start to ramp them down because they're kind of on feedback loops. And then you go out in the sun, smash, you don't even have the repair enzymes to repair it, and you're in trouble. However, Mr. Farmer goes out every day, he gets exposed to sunlight every day, and so his cells, his skin pigment increases, and the cells start to produce bucket loads of the nucleotide excision repair pathway and basic excision repair pathway proteins, and thus protecting the code.
Is that kind of like is it melanin in the skin?
Melatonin, yeah.
Melatonin, yeah. In the
It's
skin.
melatonin, yeah. Melanin, yeah. Melanin, yeah. Melanin, yeah. Melanin,
Melatonin.
yeah. Very sleepy, yeah.
So like people with darker skin typically aren't as susceptible to.
Yeah, exactly. It filters it out. And I'm going to say again, if you have dark colour skin, you are more. If you get melanoma, you're more likely to die from it. And exactly the same reason for non-smokers, because the doctor will not suspect you've got melanoma.
The thing is, people with darker skin can still get sunburn. And if you've got sunburn, then you have caused enough damage to your DNA as well.
Yeah.
So, just don't get sunburn.
If you're out in the sun all the time, but you're always wearing sunscreen, does that affect the repair mechanisms at all? Like, is that getting down-regulated, or do they still act normally?
Yeah, if you're out in the sun all the time, wear cream, which I recommend you do. Your body will be exposed to the number of hours divided by the factor of the cream. So you will be getting gently exposed to the UV, and the body will respond.
Yeah.
more timely. My argument is that people say, oh, to set the kids out, they're only going to be out half an hour in the sun. I think that's the worst time for you, because after an hour or so, your cells start to ramp up the repair proteins and start to be able to cope a bit better. But that first hour or so, yeah.
So with people that have like European like background,
Yeah.
like and now live in countries like Australia and stuff like that, is there a correlation between their like, are they more susceptible or less susceptible like on of developing melanoma because they have more exposure?
Yes, much more. Yeah. You find a lot of British people now are getting melanoma because they go on holiday to Spain for 2 weeks every year. So see no sunlight for 360 days a year, and then lie out on a beach with no skin gear on.
But we still have higher rates than they would. Like,
yeah. We have higher rates because
but that's just pure
correlation.
The amount of exposure we get.
Yeah, absolutely.
But if you were to like look at it, say like exposure time versus like rates, they would have higher rates.
You probably, if you went to exposure times versus rates, you might equalize. I don't know, but I would suspect you would equalize.
Okay.
Yeah.
Sorry, I feel really bad because I know you want to keep going. So does that mean, is there a possibility that genetically, because we're more exposed to a harsher rays, that we could potentially have more of the DNA repair genes to withstand increases in melanoma?
So, well, there's a couple of things there. Just terminology. We don't get more genes, like we get more protein. Make more protein. Like you'll respond to evolutionary. The only way that we could adapt to the UV is if there was an evolutionary pressure on bursts. Yeah. So if because we have a healthcare system, right? So we don't like people to die, with their children. And evolution tends to allow us. It's about survival of the fittest, right? That's what drives evolution. And with medical intervention, that kind has been removed from the human race to a big degree. Yeah. So we're unlikely to adapt. Yeah.
Oh, I'm so sorry. I was just wondering, like countries that have like really like up north where they're like you get a lot of sun for like summer, and then in winter they go into like basically darkness. Would they have like a higher chance of developing cancer?
So you're talking somewhere like Finland.
And then they're getting like so much sun.
Right. It's all relative, right? If you go to Finland in the middle of summer, it's like 12 degrees.
Oh, okay.
And it's because the sun is at an angle. We're closer to the equator, so we're getting the kind of the direct. The further you go away, the sun is at an angle, and therefore you get less energy. So they don't tend to get that. The big problem, so you wonder why people in the northern hemisphere have pale skin. I can't remember the name of the vitamin now. Which vitamin do you need? D, right, yeah. So you have sunlight to make vitamin D, and therefore we have pale skin because we don't have much sunlight. Therefore, we have to let sunlight in to make vitamin D. If you have dark skin and live in the UK, you have a problem because you're likely to become deficient in vitamin D, and so you've got that's quite common deficiency. Yeah. So it's just that day and night thing is just relative to it still doesn't get hot. I'm from Dundee in Scotland. We're much lower than Finland, but our average summer temperature is 16 degrees. So it's
He plays water polo
I used to do open water swimming in Dundee. All right. Okay. So and we still don't understand the role of diet in cancer. Certainly, barbecuing food, acrylamide has a clear link to cancer, but the jury's not quite there yet to understand the link between diet and cancer. Obviously, colorectal cancer can be inherited as well. And again, that's a failure of the basic system. Not basic, it's mismatch repair pathways are deficient in these people who have inherited. All right, so we get DNA damage, we can ultimately get cancer, and then it's that's bad enough. But cancers, they're no longer part of you; they're acting as survival of the fittest. I think there's zero chance we finish them, but it acts as survival of the fittest, and so each cell within that tumour is acting for its own self. And so what does it do? What it does is it starts behaving like it's a bacterial community. Bacteria don't act in each other's interests in general, other than polar sensing, but so survival of fittest. And what you find is that the cells within that tumour that are able to shift around the genetic code, maintain mutations while stopping apoptosis, are the ones that are going to survive. So if I took a tumour out of somebody from a primary site, and I took cells from each side of the tumour, that tumour might be 60 billion cells or so, and I took the sequencing and the genetic code from each one of those parts of the tumour, they would all be different. And the reason they're all different is they all came from original clones that were slightly different. Now, the part of the tumour that's pressing against the liver is in a slightly different environment than the part of the tumour that's fully exposed to the peritoneum, etc. And so there's different environmental factors, and they'll drive different types of clones to survive. And it's that kind of changes that cause what we call tumour heterogeneity, environmental adaptation, and they're the phenotypes that will ultimately, unfortunately, kill the cancer patient. Because resistance is the same. Yeah.
Just wanted to ask, if you say that about each cell within the cancer cell tumour itself, what about when we're looking at it from sort of the DNA from, say, day 1 to day, I don't know, 30 or 60? Is there going to be a continual change of that?
Absolutely, yeah. Yeah, it's continually changing.
So if you tested someone, say, at the start of their particular disease progression with them on a treatment, you're not definitely guaranteed that that's actually going to work a certain period down the track.
Yeah. So you can imagine you put somebody on, say it's lung cancer, you put them on cisplatin, which is the number one first line therapy. Cisplatin is basically a poison that poisons your DNA. It's designed to destroy your DNA, right? And it takes advantage of this. Now, the cancer cells are shutting around their genetic code, and by giving them cisplatin, you're just trying to push them over the edge that they can no longer cope. Whereas your normal cells have a bit more tolerance to cope with it. It's effectively a poison, though. You got a tumour of 60 billion cells, and the genetic code of each one of those 60 billion cells is slightly different. You only need one of those cells to have a genetic change that makes it resistant to cisplatin. And then that the tumour will disappear except for that one cell, and that one cell will grow out, and ultimately you'll end up with issues. So it's all about changing, and even things like resistance and invasion, etc., are all driven by these genetic changes. So how important is a stable genome? I was kind of touching on some of this. So it's 60 trillion cells in the human body. Genetic code of each one of the cells changed 30,000 times. And so during this hour, I'm speaking to you, 70,000 trillion changes will happen to genetic code. And in your lifetime, I don't even know what you call that number, but it's a large number of changes. And so the question sometimes is, why is cancer not more common? So here's a cool thing. We're talking about UV radiation. So, here's a British man sitting on a beach in Benidorm. Rather than wearing sun cream, probably wearing coconut oil to fry himself, and he gets burned. And you go red, right? When you get burned, you go red. Yeah. What happens the next day after you get sunburned? You heal. Yeah. Look at that. Yeah. So if we look at a cell, what's exactly just happened there? So we've got the cell, a couple of cells here. That kind of greeny colour is your DNA. Sitting within the nucleus, and the yellow and purplish colours are the cytoskeleton of the cell. Marley sitting out there in the cytoplasm, and so they're all fine, right? And then that's your DNA. These are mitotic, right? DNA. That's when the DNA doesn't look like that. DNA is usually like what you see up here. You see that? Yeah. DNA is usually spread out, but during mitosis it forms these bodies, and these pink things are the telomeres, and the blue is the DNA. It's the easiest way to show DNA. And so what happens is that man's gone out on the beach. These cells have been smashed with ultraviolet radiation. It's passed through the cell, smashed the genome, caused cross-links between nucleotides, which normally thymidines. And what happens is a biological clock gets activated. And that biological clock has seen that there's heaps of DNA damage occurred in that cell. It's worried that cell is going to turn into a cancerous cell, and therefore it sets off a biological clock. And that biological clock is timed by a protein called P53. It's not a very exciting name for a protein, it just means it's a protein that migrates at 53 kilodaltons. But we often refer to it as the guardian angel protein. The guardian angel protein here is really to detect DNA damage, and when DNA damage is too much, it has one role and one role only, and it comes back. And so after, say, 24 hours, it comes back and it finds that you've still got heaps of DNA damage in those cells. It's worried you're going to get cancer, you're going to die, and therefore it tells the cell to do, well, it doesn't want you to be cancer yet, it tells the cell to do one thing, and that's to commit suicide. So when your skin cells start to peel, just like it's mass suicide of your cells on the top layer of your skin in an attempt to stop them becoming cancerous. Yeah. So why is cancer more common when we get older? This is the risk of getting cancer in your age. So you can see that graph there, right? And from the age of about 45 or so, it starts to become exponential, the risk. The daily risk, the annual risk becomes exponential. Starts to taper off a bit when you become 80, and then other things get you. Well, let's look at this. So it takes on average, say 63 years to get cancer. 63 years, that's a reasonably long time, right? Let's have a look at mice. Why do mice get cancer? It doesn't quite make sense, does it? Because they don't live at 63 years old. They start to double from about the age of 10 months onwards. So it doesn't quite make sense, right? Until you start to think of it in a more gloomy manner, and that is cancer just a way of getting rid of us, right? Let's think of mice. They have children when they're quite young, like 16 weeks, I think they can start having their own pups. And so they have their pups, the pups hopefully grow up. Mice don't look after the kids, right? They suckle them for a bit and then they're off on their own. So you don't really need mom and dad mouse to hang around there along if you want just to pass on. You all know the selfish gene theory, yeah? It's only your genetic code that wants to go forward. And it does anything it can to go forward and doesn't really care about you in the end. And so let's look at us and like when do we have humans have kids? We have our kids probably in our early 20s to mid 20s. We take maybe 16, 17, 18 years for them to grow up and leave home, and then suddenly look, we got cancer. So the truth is we can slow down the rate of cancer by doing our risk of cancer and expand our lifespan by doing one very simple thing, and that's going on a calorie restrictive diet. That means a diet that you're hungry all the time, right? So we don't want to do it. And we know that we can shorten our lifespan by eating a lot of meat. And we know that the shortening of our lifespan in meat is down to a single amino acid called methionine. So no one take methionine supplements because that's not going to do well for you. And again, let's think back. We didn't always have a healthcare system, right? So why would meat shorten your lifespan, right? Let's think about it. So you're eating a lot of meat. You got calories are not a problem to you. The chance of you carrying your baby to term is going to be pretty good, right? The chance of your child reaching adulthood, your first couple children, 3 children reaching adulthood is pretty good, right? And therefore, let's get rid of mom and dad a bit earlier and allow the sources to be carrying on a genetic code to the next generation. Now, let's say if you're restricting calories, the chances of the mother carrying a baby to term are significantly reduced. The chances of that child surviving childhood are significantly reduced. So nature is just giving you that little bit longer to enter a time in your life when there's going to be enough calories for you to reproduce and carry on your children. Yeah.
I also heard that it's the protein synthesis that counteracts. I always call it autophagy, but
Autophagy. Yeah.
So because it's the mTOR gene that's activated when you eat meat, which creates protein synthesis, which counteracts.
Yeah, mTOR, right? mTOR is a nutrient sensor. Yeah, so mTOR is sensing if you've got lots of calories or very few calories. Rapamycin is a drug that inhibits mTOR, and rapamycin give that rapamycin, if you give that's an mTOR inhibitor, and if you give that to mice, it will live longer. There's no scientific evidence that we live longer, rapamycin, but just let's say the world's best known aging scientist, David Sinclair, he takes rapamycin. Yeah. Well, he actually doesn't take rapamycin. He takes the he takes metformin, to control his sugar. But he also takes not rapamycin. He takes no, he takes that as well. No, he doesn't. But nobody takes NAD plus because NAD plus can't transfer through cells membrane. So you take NMN or NR or NR. Yeah, I take NR.
You do.
So I'm trying to remember it's in red wine.
Resveratrol.
Resveratrol is what David Sinclair takes. And resveratrol is an mTOR inhibitor, a natural mTOR inhibitor. So mTOR, you're exactly right. mTOR is a nutrient sensor, and if you can trick mTOR into thinking that you're calorie restricted, then we know in animals that extends life. We don't know in humans. Why don't we know it in humans? Because no one will pay for a clinical trial because it's an off-patent drug. So if you pay $3 billion to run a trial to see if it extends life, then everyone in the world can make it and sell it. Yeah. So.
Well, that's what he's saying. He's trying to bottle it, but I don't know how you can bottle calorie restriction. So.
I know. But if you were able to take rapamycin legally, it might work.
Is rapamycin what they give to people that are diabetic?
I don't know what rapamycin is normal. And metformin is what they give to diabetics. Rapamycin is quite interesting because it inhibits more than just mTOR. It inhibits a few enzymes, including LQB601.
You've probably got about 1,000 slides to go.
Yeah, I'm not gonna obviously manage to finish it, but that's okay.
Sorry.
You can go online and see all the slides.
I should have waited to the end.
All right. So our interest is in children. So I said some of us can be born with the BRCA genes or the MSH1 defects for colorectal cancer. Some of us can be born with defects that are so detrimental that we will age really quickly. This is one of the syndromes that we look. It's one of the progeria syndromes, and you can see this child at 1 year look okay, but starting to age at the age of 2, 6, 12, and 32. This is a rare one. We're interested in this one because normally people who are born with these rare aging syndromes die in their teens, and they die from cardiovascular disease, including strokes and heart attacks, and they'll die of cancer, and they all. Now you think, well, he doesn't look like an old person, right, at 32 years old, because, but he's aged under a tight. We look are normal old because we've aged over a long period of time. But if you can accelerate that, you can imagine that you're not going to look exactly like an old person, but they have severe osteoporosis, they get dementias, etc. And so the reason I started my genetics company was because in Australia it was taking 8 years to diagnose these kids. And I knew that we had the technology to do it sooner. So I started a company that can diagnose them in 2 weeks from a single drop of blood.
What does that technology actually look
for like
It's
individual-
basically head of next generation sequencing.
Yeah.
Next generation. That's all we do. Next generation sequencing with our own software that analyzes the genome. Yeah.
Okay.
That's why it infuriated me, because it was already technology invented, and we just weren't training our kids right. Yeah. So these are some of the repair pathways. I'm going to flick over this quite quickly. There's like 4 main pathways. Each has distinct types of damage it repairs, but they all overlap slightly. And then that's HR. So there's over 220 different proteins that we know of involved in DNA repair, and that number increases every year as we start to understand more about biology. And so some pathways can be enormously complex. On the left-hand side there is a pathway called homologous recombination. That's what BRCA1, BRCA2, ATM all sit in. And that pathway repairs double-strand breaks, and it does it in a really cool manner. So you can imagine you lose a bit of genetic code, right, because you've had damage happen to it. And we've got 2 copies of our genetic code. So say you went to the library and you're reading a book, novel you've been looking forward to, and you found a page had been ripped out. Right, you can maybe guess what was in that area, in that page from what was before and what was after. And that's a bit what like non-homologous end joining does. It kind of just guesses and sticks it glues it back together. But homologous recombination on the left, it will actually. So the only way to get your information back in the library is go and find another copy of that book, photocopy the missing page, and stick it back in the book. That's exactly what homologous recombination does. So one of the chromosomes, one of the sister chromatids has been damaged, right? It's lost its genetic code. So what it does is it goes to its sister, it invades its sister, and now made a 4-way holiday junction, and then it will copy that bit of missing information from its sister. And once it's copied that, it cuts the holiday junction and comes back out again, and it's got the full information. Does not make a mistake, never makes a mistake. But HR, so why does not all our cells do it? Because we can only actually do HR when there's 4 copies of our genetic code present. And that reason is we could do it when there's 2 copies, right? But what's going to happen is you're going to lose heterozygosity. And that's a problem. That's ultimately a problem. So we wait until there's 4 copies of the genetic code present. So that's later in S phase through G2 of the cell cycle. Whereas the other method, non-homologous end joining, that will happen during any stage of the cell cycle, but less frequently during S phase. That's a whole other story.
Question.
Yeah.
Sorry. Is there like currently anything that's like anything you can take, or like something that'll like help kind of start that process?
That process is pretty good, right? When you're young, it's pretty good. What I didn't say was, and this is probably the most depressing thing, when you hit by the age of 40, you saw that after that, the risk of cancer became exponential each year. Our body, I haven't quite reached there yet, Sally has. Your body starts to switch over. Sally missed one.
I did it!
Your body switches off, starts switching off your repair pathways. Starts to ramp them down. And that correlates with increased mutation load and increased risk of disease.
And there's nothing that we can supplement or like to kind of bring up that process?
Yes, absolutely there is.
Like
Certainly
NAD+.
from yeah. Do you know why NAD+ works?
Isn't it one part of the powerhouse of the cell, or helps the cell work, or something like that?
NAD+ is generated by a scavenging pathway in the cell, and as we get older, NAD+ levels drop. The main enzymes that use NAD+ are PARP1. PARP1 is a DNA repair protein. It's one of the critical DNA repair proteins. So as we get older, NAD levels drop, PARP1 activity falls, and we start to not repair things.
Yeah.
The other enzyme it uses extensively are the sirtuins. Heard of sirtuins?
Yes, yeah.
Sirtuins are intrinsically linked to aging. Dropping sirtuin activity causes aging. we know that we make mutations into it, we can make mice age faster, and that correlates to any. That's why David Sinclair takes he takes NMN. NR and NMN are both pretty comparable to take, and they pass freely over the membrane of the cell, unlike NAD plus, which cannot pass over the membrane of a cell.
Yeah, I think it's just called NAD plus booster, which I'm assuming has the precursor molecule for it. That's why I take, but I think I don't know. I promote my parents to take it just because David Sinclair's research is really
I won't. You can ask me afterwards. I'll actually got a meeting after this. But I take four supplements every day. I take NMN or NR. I take glycine, N-acetylcysteine, and alpha ketoglutarate. Ketoglutarate. Keto. Yeah. It's a part of the Krebs cycle. So you can look them up afterwards and see why I take them.
Yeah, for sure, definitely.
One more, I think. We're almost out of time, though. So
I might just go to questions now, and you can see my. If you want to know anything, just email me. Yeah, I'll go. Yeah, absolutely. You get redundancy. So so the best good example is xeroderma pigmentosa patients. That's a mutation in the nucleotide excision repair pathway, or how you repair that type of damage. The basic excision repair pathway, the long patch will cover it, but it's never as good as it. So xeroderma pigmentosa people can't usually go out in the sunlight. They usually have to spend their days indoors or wear full body suits when they go out. In theory, they should die and not be able to fly.
Hey guys, listen, please.
Yeah, but because of the long patch VER can repair some of the damage, then they can survive. Yeah.
Let's have some questions from people who haven't had a chance yet, and then maybe
we'll start there and move backwards.
So with is the non-homunculus pathway you've repaired?
Yeah.
It's the one that just guesses. How does it guess?
It's probably giving it more credit. It doesn't actually even guess, it just glues things back together and hopes that there was no information lost. Yeah. So it takes two ends and just glues them back together again. Whereas,
when you were talking about the cells committing suicide and then causing the peeling after sun damage, is there a difference between causing peeling and causing like tanning? Because like
100%. So the tanning is just your skin responding. The melanocytes start churning out, and you're getting darker. So it's there. It's like saying, all right, you burn yourself, right? I'm going to make more of this to stop it happening again and reduce the risk.
Yeah, I wasn't sure because I don't tan at all and I just burn.
Yeah.
But like my brothers, they tan, which is like, I'm like, why don't I?
And that can, it's how your genes are separated. If you look for the number of melanocytes you have, you might have less melanocytes than them.
Hi, you mentioned that you have ways to advance and speed up aging in mice.
Yeah.
Are there any ways you know of that slow down aging?
We can slow down aging in mice.